So with that introduction, we invite you to
Thank you. Well, good morning, everyone. I've been here for a couple of weeks, and it has been an absolute pleasure to visit your beautiful campus and to talk to people here. I'd like to say thank you very much to Professor Tampi, who's acted as my host, and also to Professor Sen and his team, who have done such a lovely job having this nice conference to finish the visit with. I'm also very impressed by graduate students who come to talks on Sunday mornings. So I gave a talk in physics a week ago, and I managed to get the wrong title on that talk. The talk was the right talk, but the title belonged to this talk. So don't panic that it's going to be identical. The talk is going to start the same way as that, in case any of you had to listen to that one as well. But then after the first 10 slides or so, after the background, it's going to diverge. And in particular, I'm going to talk about whether you can think of cell layers as an example of collective active matter physics, as an example of an active nematic. So active matter. Active matter takes energy from the surroundings, and it does it on the single particle level. And it is meant to be out of thermodynamic equilibrium. It spends its life, and life is the right word because often it's particles which are alive, transducing energy. And so it never comes to equilibrium. And so very obvious biological examples are cells and squishy cells and microswimmers and things like we heard about in the last talk, those active polymers. If you take these systems and, is there a way of stopping this microphone? Try that better, yeah. If you take these systems and put lots of them together, so you have collective active matter, what happens is that you end up with something called active turbulence. Lots and lots of microswimmers put together in 3 dimensions here will give you these chaotic fluid flows. That's a bit strange, right? Because we're at low Reynolds number here, so why are we getting chaotic flows? If you look instead at, I press the right number of times. We'll move that one. That should start that one. Yeah. This is a mixture of microtubules, long polymers, driven by motor proteins. So the polymers are pushed relative to each other. And again, you get this turbulent-like state. And looking at cells on the substrate, again, they're moving around. And you're seeing this active turbulence. So what's going on? Can we describe this in terms of a theory which applies to all these different systems? Because being a theoretical physicist, all this detail that you engineers like to look at is too hard for us. So what I'm going to do is first of all talk about the active turbulence and tell you how nematic symmetry combined with activity can end up giving you active turbulence. So this is sort of doing the physics of what's going on in these systems. And then I'll explain how active systems are something to do with what's going on in confluent cell layers. We see active turbulence in these cell layers, which is perhaps a bit surprising because these cells often are round to start with. And then I'll talk about extensions to the theories of active nematics, which can be used to describe cells in active layers. And I sort of understand the physics and the maths behind it, but there's still a lot I don't understand about the biology. So any input would be welcome. This is very recent work. There. So let's start with the nematic. We all know about nematic liquid crystals. Nematics are long, thin molecules, and they tend to line up with each other so that you have orientational order, but no long-range positional order. This direction I'll be calling the director. It's just the director these things are moving along. And then I have this order parameter Q. Q is just an order parameter. It tells me about the ordering in this state. The largest eigenvector is this direction, and the largest eigenvalue is just the magnitude of the order. In a nematic, if you get a little thermal fluctuation and a localized distortion, that's no problem. Dead easy to put it back how it's meant to be. The elastic forces will just relax you straight away so that everything is aligned again. But in these systems, you can also get more complicated defects, ones that are more of a problem, these topological defects. And the idea of a topological defect is that it destroys the nematic order all the way out to infinity. One of these things on its own will actually have an infinite energy because you can't undo it and get back to the nematic alignment without making changes everywhere. So it's like a knot. And in 2 dimensions, the most important topological defects are this one, which is +1/2, and this one is -1/2. And in a passive liquid crystal, these defects will annihilate in pairs to give you back your perfect ordering. And in fact, they can't exist unless they're in pairs, because else they would have an infinite energy in an infinite system. Now, liquid crystals are a liquid. So just like one has Navier-Stokes equations for a simple liquid, how would you write down the equations of motion for this liquid crystal? And that's actually well known. You don't really need to know about the details, just the general form of these equations. So this is like Navier-Stokes, fine, in an engineering audience, right? Navier-Stokes, but for this liquid crystal. So you need, first of all, an order parameter an equation of motion for the order parameter, which is advectored by the flows, which turns in the shear. This is an extra bit because I have an extended molecule, and if there's a shear flow, it's going to turn. And then I have this term here, which is a mean field relaxation. The things relax down to the minimum of the free energy. And that's in there so that if I turn off the flows, it will order properly in its nematic state. And the free energy I'm going to consider is the usual Landau-de Gennes free energy, an expansion in terms of the order parameter. So these are the bulk terms, and this is the important elastic term, which says it doesn't like having gradients. And then you have another equation for the fluid flow. Looks like the Navier-Stokes equation. This is the derivative of the stress tensor. And that stress has the usual viscous stress, which looks like velocity gradients, but also an elastic stress, which says if I've got a molecule and it moves like this, it's going to induce flows in the system. That's for a passive pneumatic. We want to describe these active pneumatics. And in active pneumatics, what's different? And the answer is that these little swimmers, which are now my pneumatic particles, produce forces which act on the fluid. And very much like we heard about in the last talk, these are dipolar forces, either ones which are extensile or ones which are contractile. And the reason these forces are dipoles to leading order is just because these are autonomous particles, so you can't just have a force without a reaction because of Newton's laws. So we'll be looking at this a bit later. Remember about these dipolar forces. So we need to put those dipolar forces into the continuum equations of motion. And to do that, you have to take these and coarse grain lots of those little swimmers. And I won't have time to actually do it, so let me just tell you the answer. The answer is actually very simple. You just have one extra small term in the equations of motion. It goes in here in the stress. It's equal to zeta, which just counts the number of swimmers, and then this order parameter q. And it's an order parameter q because the symmetry of that little dipole force has this directive field. And it comes in the stress tensor. So that means any changes in the order parameter q give you a stress, all right, because it comes under a derivative. So any changes in q give you a stress and give you flows in this system. So what are the consequences of that? Well, if I have one of these little localized distortions which don't matter in a passive liquid crystal, if I turn on the activity, this is a gradient in the order parameter, so I'm going to get stresses and flows. The flows are in this direction, so they're going to make it worse. They're going to make that small deviation worse, and the whole thing gets churned up into active turbulence. What it does to the topological defects is, as well as topological defects annihilating in pairs, in these active systems, you're pumping in energy, and so you also get them created in pairs. And then they move around. They move around because, again, I have gradients, changes in the order parameter, and that gives you flows which push these defects around. So what you end up with is this active turbulence. Let's see if we can get this to play again. Okay, you can see the instabilities, the bends grow. You end up with these complicated patterns, and when it stops, we'll be able to see the topological defects quite nicely. There's a -1/2 defect with a threefold symmetry. This is the +1/2 defect. So this active turbulence is a gas of defects moving around, continually being created and destroyed. Okay, so that's a summary of active pneumatic physics. Now let's think a bit about these confluent cell layers. So what I'm talking about is epithelial cell layers. Epithelial cells line the organs of the body, and they're very important sort of tissue in the body. And they are, I always think of them as squishy cells. They tend to form layers in the body, and you can take them out and put them on a Petri dish, and what you end up with is something like this. And this has dynamics which, again, looks chaotic. And if the cells divide, so there are more of them, or they change shape, they can jam. So here they're jammed. The centers don't move very far because there's so many of them. So jamming is one possible state of this system. Another way these cells can move is flocking. They can suddenly take off and all move in the same direction. An example of this is the lining of the egg chamber of Drosophila, where the lining just moves round and round. This is what we're seeing here. And in the next movie, you'll see individual cells lining this egg chamber of Drosophila, all moving in the same direction. So you can have jamming, you can have flocking, and the other thing you can have is active turbulence. So let's look for the evidence for active turbulence. First of all, people have measured velocity fields in these cells. The velocity fields have lots of velocity jets, and they have lots of velocity vortices. One of these is a simulation of the equations of motion. One is experimental measurements. They certainly look the same, and I haven't put all the velocity-velocity correlation functions and things up there, but if you measure them, it looks the same in the cells and solving the sorts of equations that I was writing down. People have found these topological defects in all sorts of cell layers now. The next movie, which is that one. These are showing the plus and minus half topological defects moving around in bronchial cells. People have found them in maybe many different sorts of cells. Let me show you our original work on this, because it does throw up a sort of question, right? Why are cells which on average are isotropic, why are they pneumatics? Why am I calling these things active pneumatics? Pneumatics have to be elongated particles. And the answer is here that what happens is that if a cell by a fluctuation extends itself, these are squishy so they can do that, it tends to pull its neighbors and extend its neighbors as well. And so at least locally, you get things lined up in the same direction, and you get pneumatic-like order. Here's a picture of that. These are cells as a function of time. They don't move very fast. This is a line, which is the director, the long axis of the cells. You can see that locally you get pneumatic ordering, And you also get defects. These are +1/2 topological defects, which last for a long time and which move. What's more, if you count the number of defects, so counting the number of defects as a function of time, okay, what happens is that if you add blebistatin to these cells at time t is 0, blebistatin is a sleeping pill for cells, sends them to sleep, makes them passive, not active. The number of defects goes down because they're annihilating and no new ones are being created. And then if you wash out the blebistatin here, the number of defects increases again. So these really are behaving like the active topological defects you expect to find in active pneumatics. Another perhaps slightly more technical one for those that know about active matter. If you take this active matter and you confine it, you always end up with a regular pattern. The vortices can't form properly, so what happens instead is that you end up with a regular flow, and here it's a regular flow round this system like that. And that happens for macroscopic active matter as well. Have talked about the equations of active pneumatics, and we've talked about the fact that these confluent cell layers look like active pneumatics. Am I doing the right theory for these things? Okay. So I'm going to tell you some experiments which surprised us a bit, and then tell you how we might try to write down a theory which describes the experiments. This is work done where the experiments have been done by Jacob Nuckbaum's group in Wisconsin and his students Jay and Molly. And in Oxford, the work's been led by Marana Nejad, who'll be going off to a postdoc at Harvard in a month, just submitted her thesis. Liam, who's now earning lots in Germany after submitting his, and Guanming, who's at NYU now doing a postdoc. And the system we're considering, the experimental system, is one of these layers again. MDCK islands of cells. And what my experimental colleagues did is first of all measure the director which is associated with the shape of the cells. That's really what we've been talking about so far. The cells are elongated, so you measure a direction which is just the long axis of the cells. At the same time, they measured the stress axis of the cells. You can look at this substrate and you can do traction force microscopy and work out the forces on the cells. And we're assuming that these are dipolar forces, so you can identify a stress axis, and in particular, we identify the axis of maximal compressional contractile stress. So let me talk about that a little bit more. We've had this picture of a swimmer, and we said that there was a contractile dipole, and you can see for the swimmer, this is the direction of the shape, and this is the direction of the stress. They're parallel to each other, and for these swimmers, that's all there is to it. It'll always stay like that. Cells are a bit the same, right? Here's a cell, and here's the contractile forces acting on it. What we expect for cells is that if they elongate, they tend to pull themselves back to circular, which would need a contractile force. So the shape axis is along here. This is the long axis for shape, and the stress axis is along there. And that's what you expect for cells as well. But the big difference is that cells are squishy. So what could happen if you keep pushing here is you could squish them, and you could end up with something like this, because they'd be pushed inwards, which means they'd bulge out that way. And so here, it's a bit different, okay? You have a stress axis like that, and a shape axis like this, and they're perpendicular to each other, which would be perhaps a bit surprising. And so let's just define the variable I'm going to be talking about. This variable is the misalignment angle. And I'm saying, okay, let's just pretend for now we have a cell where the shape axis is in this direction, the contractile axis, the force axis in this direction, and this is the angle between them, which I'm going to call the misalignment angle. And really in cells, we expect that angle to be 0 or close to 0 because you expect cells, once they've extended, to try and squish back to being circular. So, what happened in the experiments is that we looked at this misalignment angle in the cell layer. This is something, a region from the middle of the cell layer. And the red regions have this misalignment angle smallish, actually between 0 and 45, which you'd expect. But then there are these blue regions where the misalignment angle is large, between 45 and 90. And they're not just random cells, they're correlated regions. Okay? And they evolve in time. This shows you how they evolve in time. For those of you perhaps a bit more used to cells, the red regions are contractile and the blue ones are extensile. So somehow we're getting these places where we're getting these large misalignment angles. Typically this is maybe about 4 or 5 cells across. So they're little patches of cells. Here's a movie showing the same thing, just so you can see these patches evolving with time. And what I'm going to show you on the next slide is the same thing, just with a little bit more data. Of course, it's not really a bimodal thing. The angles are not 0 or 90. There's a range. We've just taken the cutoff at 45 degrees. So here I'm plotting the probability of the misalignment angle against theta. And you can see most of them are red. Most of them are more or less aligned. But quite a lot are actually misaligned by a large angle. And so then we looked at 11 different samples and measured the fraction of red bits and the fraction of blue bits. And you can see that it's pretty much the same between different samples, which was good. In fact, very much the same, given it's a biological experiment. So what's going on? What might be happening? Well, this stuff is doing active turbulence. There are flows all over the place. And flows can certainly change the way the cells are. They can affect the cells. They can certainly affect the shape of the cells. Here's a topological defect. And there's a sort of cell picture of the same topological defect. And the topological defect is moving in this direction. It's a contractile one. It's moving in that direction. And you'll see that in front of it, the cells are elongated in this direction, and behind it, they're elongated in that direction. So the passage of defects and not only the passage of defects, but all the flows in this system can be changing the shape. They also can be changing the stress. Maybe the stress is due to stress fibers. You could imagine them being tilted or mucked up by the flows. Maybe it's due to something else. We don't totally understand that. But it's perfectly reasonable that flows change shape director and stress director. And it's pretty obvious that they change them in different ways so that sometimes they can end up at a large angle to each other, and then they relax back slowly so that they're more or less parallel to each other. So how might we do the theory? Traction force microscopy is really hard. You know, you worry about it. Okay, so these are my equations. These are not going to give what we've seen. How will I extend these equations to try and model my system? Well, first of all, we now have 2 directors and 2 order parameters. So we're going to need 2 different equations, one for the stress director, the stress order parameter, and one for the shape order parameter. Okay? And they're both going to look the same. Then we can't have that Q here. What do we do with this Q here? Well, it's the stress which is causing the flow, so let's make this the stress order parameter. The two equations are coupled by the flows, because the flows act on both stress and shape. And then we also have to change the free energy so that they respond in different ways to the flows. And what we do for this, there may be more than one way, but what we do is use a different elastic constant for the stress order parameter and for the shape order parameter. The cell's shape finds it a bit easier to line up than the cell's stress. And then, because the experiments say they relax to each other, we put a term in the free energy which allows the shape and the stress slowly to relax to each other, and we can match the time scale between the simulations and the experiments. If we do that, this is what comes out of the simulations, and I've put the experiments back there so you can compare them. We see exactly the same thing. Well, it's not exactly because it's a stochastic process, but we see the same thing. We see again these large blobs of misaligned cells. And that makes me much happier about the interpretation of the traction force microscope, because we do know what we're doing in the simulations. Of course, we can actually also match everything like correlation functions, but we do have quite a lot of parameters to play with, so that's hardly surprising. The other thing in this system, which is slightly, it was the thing we noticed first and which we worried about, is that the topological defects tend to sit very close to these boundaries where the misalignment angle is close to 45. They do that in the experiments, and they do it in the simulations. So we thought we'd better measure that effect. So we took a region close to these interfaces, which was the same size in the experiments and the simulations. So yellow is how many sites there are at an interface, and green is the fraction of defects at the interface. So two things to notice there. For some reason, the defects collect at the interfaces. I don't understand that. Secondly, there's again, with no extra parameters, simulations and experiments obey with each other very well. So how long have I got?
You're done with your time.
I've finished.
Yeah.
Right, okay. So that's my conclusions. You do get an active turbulence in these cell systems, but it looks like we have to extend the theories and we have to decouple the shape and the stress. We've got lots to do to investigate the properties of the new equations, but perhaps a more interesting question is what's actually going on in these cell layers. What actually is the stress I'm talking about? If it's stress fibers, do they really turn in the flows? If it's somehow measuring the pulling interaction between the cells, how does that work? I don't understand that properly yet. Thank you very much for listening.
Thank you, Julia.
Thank you, Professor, for the nice talk. I have just a couple of questions. The first is, you talked about earlier that nematic order in cells, like it is pretty new, you talked about it, and you showed that it can only occur in the jammed state of the system? So basically, if the cells are under local confinement,
it needs to be confluent, but not jammed.
Okay.
Yeah. Nematic order can occur in the jam state because all the cells pull each other, but to see this active turbulence, they have to be able to move. So there they are confluent, so they overlap, but they're not jammed.
Okay, thank you. And I just have one more question.
Yeah.
That's fine. So, so the time scales of relaxation of the misaligned shape and the stress directions. So how can you comment on the time scale of the relaxation? You said that the misaligned shape will relax to the direction of the stress, And can you compare that time scale to the change in the stress direction itself because of the pulling between the different cells, right? Because the other cells are putting some pressure or some kind of stresses, external stresses on this particular cell, and it's changing the stress direction?
Yes, we have measured all the correlation functions, and the time scales are all of the order of 30 minutes to an hour. Okay, and they are close to each other, and I can tell you more exactly if I actually look up the axes of my plots. Yeah. No, they have a core which gives them a scale, but I wouldn't call them multi-scale.
So you said that it's not quite clear yet what's going on, but can you give a little bit of a physical picture? Is it that from the outside, the cytoskeleton or the actin filaments have aligned because it wants to go from one direction to the rest, and then the outside world has decided it wants to deform the cell in a different way?
Yes, I mean, the shape is obvious, right, because you can actually look at it. You can think of this thing in terms of stress fibers, and certainly we find that for the aligned cells, the stress fibers are much more obvious. You can actually see them, whereas for the misaligned cells, it looks like everything is much more mixed up, but it's still it's hard to do that experiment. But it may not be stress fibers. I mean, there are other forces on these cells which are intercellular forces mediated by the cadherins between cells.
Okay.
That could be having an effect, particularly if we're thinking about dipolar forces. So, I mean, I could have just talked about that, but I'm not convinced that that's...
Did you say that?
Because if you look at that picture there, this is the correlation functions. The shape correlation function is in black. The stress correlation function is in red. This is actually, sorry, this is space ones I've got up here. We also have the time ones, and we can measure those.
Same way.
Yeah.
Thank you.
So to pick you up on that last point, there are times when the actin stress fibers can form these circumferential actin bundles.
Yeah.
When they do you know what then happens to that bordering?
Yeah, I mean, this is the thing, no. I mean, this is the thing that stress fibers would be a lovely explanation, but you also have the actin around the edge of the cell.
Yes.
You have cadherins of cells
That's right.
pulling on each other. Yeah, and it's not obvious to me which of those, how you map all those different things onto what I'm saying, which is it's a dipole.
Okay, so we'll thank Professor Julia for this wonderful talk, and we have a token of appreciation for you.